in a previous postdoc in the lab from the Lasky lab did, where we looked at patterns of specialization in the parasitic plant Striga hermanthica as compared to the composition of their host communities. So today I'll be talking about a very different project where myself, a postdoc in the Sawyer's lab, Mung Lee, and Rory have been thinking very deeply about characterizing diversity in native Mexican maize. And what I'll be talking about today is just a portion of that project. And we're in the process of writing this up, so any feedback and comments that you all have is really appreciated. And this project today is specifically about linking root physiology to environmental associations in Zea mays landraces. So I don't think I need to spend too much time explaining to this group what a landrace is, but I'll briefly mention that landrace diversity is thought to represent fitness to environment and the local environments under which they're cultivated. So the environments in which landraces are cultivated are generally considered stressful by modern day agriculture. So there's this idea that they're harboring beneficial traits and alleles that might not be present in elite varieties that are grown and other breeding lines. And there's this real interest in uncovering the basis of this fitness, but it's coupled with a challenge of characterizing the diversity. And oftentimes when people are thinking about landrace diversity, they think about, there's a real focus on above ground traits, whether it be cob-related traits or flowering time. But in the Sawyer's lab, we like to think about how heritable differences and variation also exist in below ground traits, including aspects of root system architecture and anatomy. So there's been this real push where we're thinking about roots as breeding targets for enhancing resource capture, lessening the need for agricultural inputs, and improving plant adaptation to stressful environments. And just to illustrate a couple phenotypes that are being thought of as potential breeding targets for incorporating these or addressing these benefits, there's been this established phenotype of steeper roots as potentially adaptive for reaching water that's available lower in soil, so very helpful for a drought limiting environment. And there's also been a phenotype for architecture where more shallow roots may be adaptive for low phosphate soils where inorganic phosphate is limiting and the plant can better forage in the above or the upper levels of the soil column. And beyond root architecture, aspects of root anatomy can actually be really informative for understanding how plants are coping with stress. So just to introduce you to a couple anatomical features that we'll be later discussing and thinking about their implications, root cortical parenchyma, or these huge air spaces that are seen in the cortex, reduce the metabolic cost of soil exploration for roots, and they're thought to contribute to drought tolerance and improve nutrient acquisition in low nutrient soils. And then also metaxylem vessel size, or these vessels that conduct water up to the rest of the plant body, are going to have really large implications for the axial water transport of roots, where smaller metaxylem vessels have been associated with drought tolerance and are thought to be used as a strategy to reduce water conductance in water limiting areas. But although roots can be very helpful thinking about how plants are coping with stress, they can be really limiting from their below ground nature and the manpower that's needed to dig up roots and either ablate them or hand section them to then gather anatomical features and think about the associations between environment. So because phenotyping roots is still a really major obstacle, this really increases the value of existing data sets, and also using a modeling approach where we can uncover associations or associations between various pieces of information, whether it be phenotypic, genotypic, or environmental, and then thinking about what these associations between these pieces of information are saying before we grow many plants. So we can be a little bit more specific with our questions before we go through the huge phenotyping effort. So given that landraces are thought to be this combination of long-term environmental signals, where environmental differences are thought to in part drive variation that we see in traits, can trait values be predicted for environmentally associated landrace phenotypes? And how can we use a modeling approach to elucidate the underlying environmental gradients that might be driving trait variation? And ultimately, how can we use landraces as this long-term experiment on what might be aiding fitness to limiting environments through these environmental correlations? And what can we learn from landrace root diversity as it relates to environment? So just I'm going to quickly go through my workflow to give you guys an idea of the roadmap for what we'll be talking about today. I will spend more time talking about our methodology and our results for each section. But for our general workflow, we combined phenotypic and environmental information to identify environmentally associated traits. Once we identify both the environmentally associated traits and the aspects of environment that describe significant variation in a trait, we're able to plug that information into a model where we can predict how trait variation changes across combined environmental associations. Once we have a constructed model that's effectively telling us how we predict a trait to change across environment, we can then predict traits for other panels of georeferenced landraces where we only have environmental information and we don't know anything about their trait values. The panel of landraces that we predicted traits for was genotyped. So then we plugged in the trait predictions and the genotypic data to see if there were any top significant genes or SNPs that were underlying underlying the variation that we predicted using our random forest models. So altogether, this pipeline is working to identify putatively adaptive traits, thinking about the environmental signatures that are predicted to be involved in that trait variation, and then if we can see any of the genetic underlyings or genetic underpinnings of that variation. So the first step was taking previous anatomical characterizations by the Lynch lab here at Penn State, where they had this really large phenotyping effort of a panel of landraces that they selected from landraces antiocentes that they selected from stressful environments, and they grew them in a common greenhouse environment. We subsetted those individuals to only include Mexican maize landraces, so only including anatomical characterizations of landraces that are from maize's point of origin. But we also do have information on these landraces, architecture, and above ground biomass, but I'm going to be focusing today on the root anatomical traits for the sake of the story that we're telling. And just to illustrate the aspects of anatomy that we were interested in and that we ran through this pipeline, from these kind of images that they collected from this phenotyping effort, we were able to gather information on overall root cross-section area, the steel area of the root cross-section that is dedicated to cortex, and then specific parameters that were cortex related, including the parenchyma area, how large a cell is in the cortex, how many cells are in a file in the cortex, and then also metaxylem parameters, including the number of metaxylem vessels, total vessel area, and then also specific vessel parameters, including the diameter of a vessel and total area of a vessel. Once we had that phenotypic information for these georeferenced landraces that were grown in a common greenhouse environment, we also extracted over 100 environmental features that are representative of an accession's point of origin. So this is including information on precipitation, soil, temperature, aridity, etc. And we got this from public databases, including Chelsea and Soil Grids as a couple examples. So what this looks like is for a given accession, we have information on its phenotypes whenever it was grown in a common environment, and then its point of origins, environmental gradients. And before we hop into the modeling, I wanted to illustrate that there is phenotypic variation within this diversity panel of Mexican landraces, and also illustrate the value of this method where we're thinking about combined environment rather than individual aspects of environment. So when we do a basic phenotypic PCA of the accessions, not including any environmental information, we can see that the individuals are principally loading based off of size on dimension 1 of the PCA, where these PC1 loadings that are extracted for every individual, when they're plot against cross-section size, we see individuals that are loading negatively on PC1 are larger in cross-section size, whereas these individuals that are loading in the opposite direction are generally smaller in cross-section size. And if we plot these PC loadings against information from the point of origin of these accessions, we can see an inverse relationship between dimension 1 and elevation, where individuals that are from lower elevations generally have larger cross-section sizes, and then the inverse is true whenever we go up to higher elevations. So this is maybe helpful, but less exciting. And what about more subtle traits, such as metaxylem features? And depending on the phenotypic PC axis, we recover that there are different phenotypic loadings. So we see when we plot PC2 versus PC3, we start getting a little bit more helpful information in how we can see variation within this burden panel that exists for less obvious traits. And so what we did to explore this phenotypic variation, and if we could see generalized groupings within the burden panel, was split this data based off of the loadings of dimension 2 of the PCA into 3 quantiles. And we can see what information dimension 2 is capturing. Based off of these box plots, which just show within the groupings, these blue individuals, this blue grouping of individuals is generally having more metaxylem vessels that are larger and less parenchyma area. And then the inverse is true for these individuals that are loading in the opposite direction. And here we have images that are representative of these groupings where we can see that these blue individuals are having, in fact, larger metaxylem vessels with more metaxylem vessels than these individuals that are loading in the opposite direction. So we went in thinking that maybe this variation is related to water availability. And here I just have a basic scatter plot of a very similar idea for what we saw, root cross-section area versus elevation, where we plotted these PC2 loadings, which are separating out metaxylem vessel area and number of metaxylem vessels versus annual precipitation. We don't really see anything too obvious here. There's this very weak correlation where maybe these individuals with larger metaxylem vessels are in areas with lower precipitation, but this is really unconvincing. And this is just to illustrate that correlating phenotypes to individual aspect environment isn't difficult and likely an oversimplification of what's actually happening in nature. So this not only supports our idea of thinking about combined environment relationships to traits, where we can hypothesize that including more environmental aspects would strengthen our signal. But also, this brings on the question of how do we accurately identify the aspects of environment that are describing variation in trait? Because maybe a piece or an environmental feature that we thought was correlated to a trait is actually not so informative. So how do we identify environmentally associated traits? Because one, not all traits are going to have significant variation described by environment. And then two, not all aspects of environment are going to describe significant variation in these environmentally associated traits. So this brings in the use of a feature reduction method that we use called the Baruta method, where we are able to reduce the number of features and identify which aspects of environment are actually describing significant variation in a trait. And a byproduct of this is we also identify traits that actually have significant variation as associated with environment, or as correlated to environment, I should say. So what this looks like is for a given trait, we're able to identify if it has environmental association, and if it does, which aspects of environment are actually describing variation. And then only these final environmental features are used for model training, which is helpful for preventing overfitting because we're not putting a bunch of useless data that ends up just finding a bunch of patterns in our data that don't actually have biological relevance. So just to remind you of our 16 traits of interest, these are all the anatomical traits that we tested for environmental association. And of the 16 traits, we found that 9 had significant variation described by environment through using the Baruta method. I'm not showing what aspects of environment are associated with this phenotypes here, and I'll show an example of that later in the talk when we focus on one trait. But there are a couple interesting patterns that we can see from the Baruta method finding significantly environmentally associated traits. One, we're really good at picking up traits that have association to environment when they're proportions. So we can see that with the percent of cortex, and also looking at stele area over root cross-section area or total cortical area. And then two, these metaxylem features are coming out as having significant relationships to environment. So now that we've identified the traits that have significant variation described by environment and the aspects of environment that describe variation in those traits, we can put that data into a random forest model where we have this outcome of trait-specific models that describe unique environmental combinations associated with trait variation. And we effectively have a model that predicts how our trait changes across combined environment. And we do this for our 9 phenotypes that have significant variation described by environment. And so this is what the percent variation described by the random forest for our 9 focal phenotypes looks like. And we can see that the prediction strength really changes across the phenotypes, where we have 33% of the variation of parenchyma described by the model, but then median metaxylem vessel diameter has actually negative variation described. So unexplained variance can be due to just randomness in the behavior, random behavior in the data, or lack of fit. And what we're starting to think also is that potentially the inability for a model to pick up higher variation just could be due to plasticity within a trait also. Because if the trait is being grown in a common environment and it's generally very plastic, we're not going to recover the same relationship that it would have with its local environment. So let's zoom in on a parenchyma area, the phenotype that we could predict the most variation for, and see what these model outputs actually look like. So just to reacquaint everybody and recenter us before we look at what these model outputs look like and we start doing some interpretation, our random forest is predicting phenotypic variation using environmental information. And for a parenchyma area, our feature reduction method, the Baruta method, selected 3 environmental features as describing significant variation in a parenchyma area, including base saturation of soils, calcium carbonate of soils, and photosynthetic active radiation of winter months. This importance on the x-axis can be interpreted as how often a variable helps the model correctly classify the data. So base saturation was associated with having the most correct model classification. And even though base saturation has the highest importance, it's not necessarily the direct cause of variation. And rather, because of what our method, we're picking up the combination of these environments scales up to have our predicted phenotypic variation. And now here's a scatter plot of what our observed values are for a parenchyma versus the y-axis of what our model is able to predict a parenchyma based off of these 3 environmental features that describe significant variation in this trait. So we can see that our model is consistently underestimating the extremes, but it is picking up trends in the data, which is very promising and exciting, where we see that individuals that observe to have lower parenchyma area are actually being predicted to have lower parenchyma area, and individuals that have more parenchyma area are also predicted to have higher parenchyma area. So although we're not capturing the extremes with our model prediction, we're picking up general trends in the data to say individuals that have lower parenchyma area are actually predicted to have that, and the same is true with individuals that have more parenchyma area. So once we've constructed a model that's representative of how our trait changes across combined environment, we can predict phenotypic variation for individuals that we don't have any phenotypic information for, but are georeferenced, so we know aspects of their home environment. And in this case, we used a genotype panel of landraces that have been used, that we used a subset of genotyped CIMMYT landraces. This is the same foam panel that has coverage all the way down to South America that I think probably a lot of people here are familiar with. So once we've had this constructed model, we can put environmental information and predict how this phenotype would change across space. And in this case, we'll stick with our example of a parenchyma area. And what we see is that our random forest model predictions actually do follow the spatial patterns that are present in our observed phenotypic data set. So just to point out a couple features that are important here. One, our parenchyma area is predicted to be lower on coastal regions within our it's observed to be lower on coastal regions, and our model is also predicting that coastal regions should have lower parenchyma area, whereas more drought limited areas and highland areas are predicted to actually have higher parenchyma area. And then a really cool application of this and fact within this, since we're predicting phenotypes from solely environmental data, is we don't have to have coverage within an area or predicted phenotypes to be able to predict that region. So we can see we have no individuals that are observed within the Yucatan, but we're able to predict that we think the Yucatan would have higher parenchyma area as compared to more southern areas. And then using these trait predictions, since I mentioned earlier, this panel of about 2,000 landraces had genotypes, we can use those trait predictions, which are almost thought of as like a composite environmental score, and genotypic information to identify enriched genetic regions that may be associated with that predicted trait variation. So importantly, so you can see here, whenever we plug in our trait predictions, we get this peak on the beginning of chromosome 2. And And importantly, we actually get different signals recovered from our trait prediction than whenever we plug in the individual aspects of environment that were used to predict that trait in an EG-WAS. So you can see here, we have Manhattan plots for the 3 aspects of environment that were used to train our ANCOMA area: calcium carbonate soils, base saturation, and photosynthetic active radiation of winter. And we don't recover that same SNP early in chromosome 2 for any 3 of these environmental EG-WASs. So we're getting a different outcome. We're getting a different signal from our composite environmental score trait predictions than we would if we looked at any one aspect of environment. So now we've established this pipeline as being useful for identifying environmental trait associations, and then considering what genetic regions could be associated with that trait variation. But what can we learn about plant physiology from these anatomical trait predictions? And if you think back to the predicted cross sections I showed earlier in the talk, they aren't just pretty figures. They can also be used to estimate hydraulic properties of the root system. And this is because root anatomy defines the baseline for root radial hydraulic properties. And specifically today, I'll talk about radial conductance, which affects how quickly water moves from the cortex into the stele. And this step of water moving into the stele from the cortex is thought to be one of the largest constraints on plant water uptake, if not the largest constraint. So using our random forest models, we predict that individuals from these more northern regions and highland regions have higher radial conductivity. And we're thinking that this might be a water uptake strategy because, like I mentioned before, low radial root conductivity often limits plant water uptake. So if you have a higher radial conductivity, then you might be using that as a strategy to deal with being water limited. And excitingly, we recover a very similar relationship between this predicted KR value and an aspect of anatomy within our models, as previously characterized by Adrian Heymans, who saw that the ratio of stele to cross-section area, so how large the stele is and compared to total cross-section area, is highly tied to this simulated radial root conductivity. So we're thinking that maybe water limited areas are predicted to have KR. And this is because of the anatomical characteristic where they're predicted to have a larger portion of their cross-section area taken up by the stele. And so one of our next steps is actually validating these predictions using the founders of a multi-parental mapping population. So our lab, and in collaboration with many others who are here on the call, is making a multi-parental mapping population that whose founders are representative of diverse germplasm that span environmental gradients. Here, I'm showing diversity in the soil environment of these 8 parents, but you can imagine that just like them spanning different silt content and cation exchange capacity of soils and loading differently for soil environments, they're also representative of diverse altitudinal gradients, precipitation regimes, and temperature gradients. And so using these parents, we're going to phenotype them and see if our predictions actually align with what we see in reality when we grow and phenotype these plants. And something that's really exciting about where these parents originate from is, if you'll think back to our sampling with our training data, we didn't have any individuals that were from the Yucatan, but one of our MAGIC founders is from the Yucatan, so we can see if those predictions actually align with what we see when we grow Naltel. And then, of course, anatomy is in the context of different architectures, both root and shoot. And we recover that our 8 MAGIC parents actually have very different simulated architectures after day 28. This was done in collaboration with Adrian Heymans, who I mentioned earlier did the KR modeling and found that root cross-section size and stele proportion is highly related to that KR value. And so we're also thinking about how architectural differences can inform us about these mechanisms for survival in limiting environments, even though today I did focus on anatomy. So we're thinking broadly terms. We're thinking in broader terms also about how just whole root architecture can scale up to inform adaptation to these limiting environments. And then one last collaboration that I'll mention, and is another example to highlight success of this method, is we collaborated with a researcher, Peng Yu, who presented in Zevolution a couple weeks ago on his work on microbiomes and seminal root number. And we were given over 100 seminal root number values that were measured for Mexican landraces that we plugged into our random forest pipeline and recovered a latitudinal climb in seminal root number of Mexican maize. So we can see here that based off of these predictions, which are in green, and also observed values, Mexican maize produces more seminal roots in equatorial regions. And we were also able to identify environmental features that may influence seminal root number, as shown here on this importance plot, again, that's very similar to the one I showed you previously in Arankama area. And we recover the absolute latitude is the most important aspect of environment, but this might be a proxy for other environmental features such as pH of soils or precipitation. So here we just wanted to highlight that this method has worked whenever we have more than 39 individuals. We have 128 in model training, and also to highlight a collaboration that's come out of the maize community. So in summary, we used random forest models to associate root anatomical variation and environmental gradients. These constructed models were then used to predict traits for a genotype panel of landraces. Using trait environmental associations, we can then consider the physiological mechanisms to cope with that landraces are using to cope with environmental stress. And applications of this pipeline, there's a couple, but I'll highlight two here. One, we can use it for hypothesis generating, where we can think about if something comes out of a random forest prediction, do we actually expect a Arankama area to be affected by base saturation, or is base saturation of soils a proxy for another environmental feature that we're going to predict affects a Arankama area? And then two, guided sampling for phenotypes of interest. If we recover that there is a large spatial, or there's a large difference in a phenotype as related to two different locations, we can be really specific in where we're picking individuals to phenotype to then think about that trait of interest in the context of environment. And so ultimately here we're using landraces as this long-term experiment on diversity through environmental correlations. And with that, I'll say thank you. Thank you to all members of my lab, in particular my advisors, Rory and Jesse. Thank you to our collaborators, Adrian for making the root system architectural models, Guillaume, and then Peng for supplying the seminal root data so that way then we could illustrate that our pipeline actually works with more data than 39 accessions. And there's many more members of the lab that are new, but we don't have updated pictures. So thank you to all the newer lab members also. And with that, I'll take any questions. Thanks for listening.
Okay, thank you very much, Chloe. I propose that we start with the question of Germano.
Okay.
So I don't know if Germano, you want to turn on your microphone, or should I read your question?
Yes, thank you. I missed the beginning of the presentation. So before
Sure.
that, congratulations for your good work. What soil variables did you use it? And did you consider using bulk density and also other variables related to the soil mechanics?
Yeah, that's a great question. So I didn't list all of the variables we use because it can be a little overwhelming because there's like over 150 of them. But we do have those soil mechanical properties. Here, let me go back to the slide. So we used soil grids, which has a couple of those mechanical properties. And then we also use this global soil data set for use in earth modeling systems. So that includes information on pH, and then also bulk density and silt, so soil composition, and then also different aspects of anion exchange capacity and et cetera. Like there's probably over 30 different soil variables, but we found that precipitation and temperature were much better at describing variation in the traits.
Yeah,
but we did include them. And it doesn't mean that they're not related. Because something like latitude, if we recover a relationship with latitude, that could be a proxy for some soil variable that changes across latitude.
Yeah.
So everything's pretty correlated.
Yeah, I know soil grids is a very nice database. Also some, I don't know, temperature and precipitation are very interpolated and also very related to latitude and longitude. So That's why I was asking you if you have considered using the actual soil moisture, just like the end result of some water balancing in between the precipitation and soil properties, because I don't know, perhaps this more capture more sign than precipitation.
Okay.
I don't know. That's just a question and also suggestion.
Yeah. No, I guess that is like kind of a shortcoming with these databases. A lot of them are very interpolated, but I think with the databases that we use, we capture a lot of the variation. I think it's probably as representative of the soil environment as we could be, unless there are other databases that we're unaware of. But between these two, we capture a lot of information about soils.
Okay.
Thank you.
Thank you, Herman. And Ruben, would you like to ask your question?
Sorry, Chloe, just a technicality. So I was curious, does water radial conductivity measure just uptake from soils into the xylem, or is this a measure of the net balance of water from the soil into the xylem, xylem into the soil?
Yes, so I believe it is the potential for water to be uptaked from the cortex and for it to move across the suberized Casparian strip. But that's a technicality that I need to look into more to be more specific with the story that we're telling. But I believe that's correct. Rory might also have a more.
Or Johannes, if you're in the call as well, maybe he knows.
Yeah,
I can briefly say that we look at axial conductivity as well. And obviously you have to combine these things to estimate actual water flow through the plant, along with taking into consideration what's actually happening in the shoot.
Right.
Also, Adrian has done very nice work to show that obviously something like radial conductivity will change as you move up and down the route. And a lot of things that are going on with maturation, etc., will greatly impact how this is all going to work. So I think there's a lot of room to continue to explore these types of models to really look more closely at what's happening.
Yeah, that's a great point. Is depending on where you look in the root, you'll recover a different radial conductivity value. So younger tissues have higher KR than a more mature tissue. So thinking about where we're sampling is also going to really affect our answers. Where in the root we're sampling is going to affect our answers.
But I guess that at the moment you have not integrated that information that I guess is in the models with soil profiles and how that might change with soil profiles. I guess that's very complicated.
Yeah, we haven't integrated that information yet, and we need to think about how we could do that effectively. But that is something that I don't know if Adrian's looked specifically at how soil profiles affect KR across the root as much as he's modeled that it does change across the root system. But yes, the soil profile that the plant's growing in is going to also affect it, and how much water is available wherever in the soil also.
And just perhaps a comment there is that one of our motivations for doing this type of work is to parameterize downstream structural functional models where we can actually see how these root systems are performing, at least in silico, these types of things. And then, yes, we can vary water and other resources in the soil, etc., how they impact what's going on.
So I have a question myself. Well, two questions is like, so if I understood correctly, so because your that your environmental variables are correlated, right? So you take this into account to actually predict the different phenotypes. But I also imagine that your phenotypic variables are also correlated. And I understood that you performed a trait by trait analysis, but I was wondering how much redundant is this, given that maybe the traits are correlated? I don't know. Are they correlated?
That's a great question. I think I haven't looked specifically at correlation between traits, but what I can say is when you extract the environmental information that is describing correlation in traits, you can generally get a pretty good idea of how similar traits are based off of what aspects of environment are describing variation. Because if there's two traits and like the same 5 environmental variables are showing up, that is, in my mind, kind of answering your question where I see, okay, these two are probably pretty correlated. But I would say in general, with the traits that we're looking at, we're capturing pretty different aspects of anatomy. Some are correlated, but there's also subtleties where a trait might be correlated in one area and then it's very different in another environmental space. And so thinking about how these traits scale up to affect whole plant physiology, presumably they are going to be correlated in a way, but I think also the subtleties of where they're not correlated is helpful for answering how are these different plants using different methods to cope with stress.
Okay.
Yeah.
Okay, cool. Yeah, that was pretty clear. And then I have a second question is like, so you showed that they measured those traits in a study, right, by Burton. And I was wondering whether they measured also any trait related to fitness in that study, or, I mean, is there any evidence that those traits are adaptive?
Sure. So here, let's go to that slide. So I guess it depends on what traits you think are representative of fitness. So they measured root dry weight and shoot dry weight. And I don't think they had any kind of more qualitative fitness scores, though. Everything was very much, this is how large the plants were, this is how much space the root system took up, and then these are the anatomical characteristics. So I do not believe there was any kind of fitness value. Although I guess there are a lot of people who use dry weight of shoots and roots as fitness. So depends on what your definition of fitness is, I guess.
So when you will be looking at so I understood you will be looking at the mapping population, well, the Mexi-MAGIC, so the population, right? That are,
yes, the
so,
parent of the population.
Yeah. So you will be also mapping those traits using these populations, or?
I think so the way we're looking at it right now is more of a validation set to see, okay, so we predict an individual from this location to have this size parenchyma area. When we grow null-tel, do we also recover that the one from the Yucatan has?
Okay, you will not use crosses to validate like the locations, like, you know, the GWA that you run, the EGWA that you run. You will not use some kind of population to...
That's an idea later on.
Okay.
So the mapping population, Rory might be able to speak more to this. We don't have enough families right now to map at how fine we would like to. So right now we're just growing the parents as a validation set, but later on the idea is whenever we grow the MAGIC population, we'll be able to break up population structure and identify more specific genetic areas and see if that aligns with our EGWAs. But that's kind of a later on,
okay,
student.
I mean, something we do have more is now we have predictions of root phenotypes, for instance, for that CIMIC foam panel.
Mm-hmm.
So there we do obviously have field data, which came out of the SEEDS project. We have drought trials, etc., from that project. So we can certainly go in, and we have started to go in to then see how those phenotypes that we're predicting relate, for instance, to those types of field data.
Mm-hmm.
So again, you know, we can certainly improve our models with more training data, including genetic information, etc.
Mm-hmm.
But I think that's an idea that we then have the ability to at least come up with a predicted root system for all of these things that have already been evaluated. As much as trying to go out and then do our own trials, which obviously is pretty intensive work to try start getting.
Yeah, I'm sure. I was just trying to imagine if you had any population where you could have fitness related data as well, and then you could maybe relate some of these traits to fitness.
Oh yeah, like, I would say the best one is
okay.
So it's not when we're all our root stuff at least is projected on there, but we can at least go there and then that first cycle of modeling, predicting, looking for those colors, and that's starting to look pretty interesting.
That's super cool. Ed, your turn.
Yeah, no, so really beautiful work that you all are doing. I was just wondering, did you just look for enrichment on the GWAS side for like root expressed genes?
Yeah, so this is the work that Mung did, and unfortunately she is driving right now, and I think she's here via phone call, but I will say through Maize GDB, we were able to see that whenever we did this pipeline with a seminal root number, we actually recovered a SNP that was in a gene that was that through expression analysis was related to a root related gene. So I don't think we've done that yet with the parenchyma area, but we have recovered that same SNP using different data sets of the same phenotype. So we did it using the Burton data set for seminal root number, and then Pung's data with seminal root number, and we recover the same top SNP that's within the same root related gene.
And I would say that we're very excited. A lot of these new single cell data sets that are coming out for root anatomy. And again, that's a really exciting thing to mind when we have these types of traits to look at. And I would say there's at least one nice study that we've not talked about here where people have used inbred panels. They've used the Wisconsin diversity panel to map these traits as well. So we have a third sort of section of information that we can bring in terms of cross-referencing all these different GWAS and eGWAS and environmental type signals. So that again is sort of part of the fuller view here. What we haven't got at the moment is any one data set where we have everything.
Right.
You know, we have either genotypes and environments, so we have traits but no genotypes, or we have inbreds where we don't really have an environment. Right. And so we're kind of trying to pull together all of these things, but the logistics of simply going out and taking these traits from a very large panel of genotype landraces, etc., would be at the moment, I think, the thing. But I think there's still a lot we can explore with what's out there, as Chloe says, giving value to these existing very hard-won data sets.
Hermano, you had another question, I think.
Yeah, I was wondering if you have any other trait, no root trait for just like for example, the biomass shoot.
We do. We have biomass. So we have nodal root weight and then total, I think it's nodal root dry biomass. And that is one of our significantly environmentally associated traits. But since this is anatomy talk, I didn't talk about it. But we do have other biomass related root traits.
Yeah, because I was wondering, these root traits sounds nice, but there's any relation between these traits and the shoot biomass that you can see in the literature or your database?
So you're saying between these traits that we're talking about in the shoot biomass?
Yeah.
I haven't looked specifically at that, no. But I'm sure there are plenty of studies that compare a parenchyma area as a strategy to increase biomass in the case of drought or low nutrient. But I haven't looked specifically at how these traits relate to above ground biomass, no.
I mean, again, Jonathan has done some of this, at least in RILs and NILs. So we have from inbreds indications of which traits may be important in certain certain stresses. So at least, again, we can see if we see concordance with what we're picking up.
Stephanie, when your question, you want to ask your question?
Yeah. Hey guys, good to see you again. My question is probably, I'm just going to sound like Jonathan asking this, and I'm very sorry in advance, but we know that oftentimes that single traits or single root phenes are not maybe the best predictors of relationships with the environment or stress resistance and or stress responses, and maybe it's actually combinations of multiple traits that are better predictors of resilience. So do you have any plans to expand this workflow to kind of account for that, or just kind of curious what you're thinking there?
Yeah, that's a great question. I think we kind of see that come up whenever we go through the feature reduction method. The fact that we are able to recover these proportional values are easier to predict with environment. So that probably supports your idea that, or what you're saying makes you sound like Jonathan, where it's like these combinations are what's important. And so we haven't talked specifically about how we would address that within the pipeline that I mentioned, but I think by looking at the combinations of traits in the cross sections that I showed you here using this program, I didn't say what the program's called, it's called Grinar. I think we're starting to really address that question where we're scaling up and thinking about these individual parts of anatomy that we can predict, and then thinking about how those scale up to actually impact fitness and physiology. So I haven't thought too much about how I would integrate that within the earlier part of the pipeline, but I think we start to see that come out whenever we find what traits actually have environmental prediction. And then also when we're thinking about how those scale up to impact whole root predicted physiology.
Yeah, because I was just thinking, you know, looking to your specific example of how one principal component described this relationship between metaxylem vessel area and parenchyma area.
Yeah.
And how, like, I don't know, maybe like plugging that PC into your random forest model may account for that. Yeah, I wasn't sure, but it could be really interesting.
Yeah, that's a great idea. We haven't plugged in the PCs into the random forest model. What we have done is plugged the predicted phenotype PCs into the EGWAS. And that's kind of hard to find a signal. But that's a great idea. Yeah, I'm going to try that, plugging the PCAs into the random forest. Thanks for your question.
Okay, I don't see any more questions, but I think it was a really great discussion here. Oh, no, there is a question by Ivan. Sorry, go ahead, Ivan. Sorry.
Hello.
Hi, Ivan.
It was a very nice, hi, how are you? It was a very nice presentation, Chloe. It's always very exciting to hear about local adaptation in roots. And my question was related with the input data that you start from Burton. For example, the Burton grow these plants in the greenhouse. And we know that when we grow the plants in the field, even in the well conditions, there are a lot of difference, like in the plasticity of the phenotype. And when you put some stress, there is even more plasticity, right? So I am just wondering if you consider these plastic responses in the random forest model.
Sure. Okay. So there's a couple parts there, and I don't want to miss any of them. So one is plasticity in traits. So I think we don't account for plasticity within a trait, but if we can't predict a trait, we assume that maybe, or we don't assume, but we're predicting / thinking maybe that is due to plasticity within a trait. And then two is the idea that plasticity changes across environments. So an assumption here is that the plants that have more parenchyma in the greenhouse would also have more parenchyma in the field, and more parenchyma, and then even more parenchyma under stress. So we're overall recovering that a plant that has a larger or smaller phenotype in the greenhouse would be representative of what we would see in nature, which isn't perfect, but it's kind of what we can use given the data that we have. Okay, I think I addressed both those questions. I don't know if I answered your question completely, but it's definitely like it's not necessarily a shortcoming, but it's just something that we have to deal with. But no, I don't account for plasticity within the random forest.
Yeah, I think it's an extreme assumption, right?
Because, yeah, so it's a pretty extreme assumption. But yeah. I think it's something that people generally do. I mean, when we grow plants in a common environment, and then we see which one has more of something and less of something else, we're assuming that that's representative of what we would see normally.
Mm-hmm. So Ruben is asking Edwin to comment on that, or? I don't know.
No, I just sent you a private message maybe to ask Edwin if he wanted to add something to his comment. That was it.
Okay, I see no further comments or questions, but I think the discussion was really cool. So thank you so much, Chloe, and bye everybody.